even though this year has been marred because of the COVID-19 pandemic, but the good work that me and my colleagues are doing continues, and we're continuing to advance the field. And on that end, I think we had several updates and new data being presented this year, throughout the year at many congresses, actually. But I think at ASH, we were very encouraged to see several CAR T constructs coming to the fore. We saw updates on several BCMA CAR Ts that we were already familiar with, like ide-cel or BB2121, the J&J 4528, which is called cilta-cel now, the orva-cel construct with JCAR. But then we also saw the Allo-CAR-Ts targeting BCMA being shown in phase 1 studies, along with a very interesting transposon-based BCMA CAR T construct by Poseida. So there were several of these CAR Ts that were being presented. We also had another construct, CT053, being presented with a shorter vein-to-vein time. So bottom line is, I think for the BB2121 construct, we find longer follow-up without any new safety signals. We do recognize that some of the earlier BCMA CAR Ts are perhaps not giving a really long PFS, or we were hopeful that this may be a curative strategy. That's not the case, but nevertheless, you are getting close to a year out of the early BCMA CAR Ts in clinical trials. I was most impressed with cilta-cel because of the depth of response and the 12-month PFS that we're seeing at this point. Judging by the PFS trends, it appears that that the PFS of that construct may get to close to 2 years, but we will have to see. So I was very encouraged from on that end with the CAR Ts. The Allo-CAR-T option would be ideal as an off-the-shelf CAR T option, but the preparative regimen does involve knocking down the immune system quite a bit, including one of that company's own, CD52 directed antibodies. So I'd be keen to see what would be the downside of that. How much efficacy are we compromising? How much infection and side effects are we taking on by making some of these concessions? On the same end, the Poseida construct, very low CRS rates with that construct, but the activity wasn't as robust, with the early cohorts compared to what we would have expected, knowing what we know about ide-cel and cilta-cel.